with a segment of genes. And also from the beginning it has the operator.
Right. So an operon is a system where you have several genes share. Usually these genes are sharing a similar function, right? They're involved in, like, for example, the metabolism of lactose. They share a similar function, and so it's efficient energy-wise for bacteria to basically group them together and have them share a single promoter region. That way all these genes can be transcribed at once using a single, minimal transcription factors, minimal gene activation in a sense as well, right? So what's the basic structure of an operon? Do you guys remember that? The basic structure for an operon?
It has the operator.
But what's first? What do you usually have first?
It's not the operator or the promoter.
Operator is not first. What's first? So what is RNA polymerase binding to?
It binds to the operator.
Promoter?
The promoter, yes, the promoter, yeah.
That's what the operator is upstream of the promoter.
Operator is upstream of the genes.
Yeah.
Downstream of the promoter. The operator is actually between the promoter and the genes. And there's a reason to that. And then now you have, if we're talking about, let's say this is the lactose, right? The operon. We have lac Z, lac Y, lac A. Right? So this is like the basic structure. You have promoter, right? One promoter for multiple genes. All these are genes right here, right? Genes right here. And so they're sharing this one single promoter that's going to be basically result in a transcript of mRNA that's going to have all these genes on it. And so the operator is what? What binds to the operator? The operator is a regulatory function, a regulatory region of like the operon system in prokaryotes. And it's responsible for, as like a backup safety mechanism for regulation, where in case you have RNA polymerase bound to the promoter and it's going to go ahead and transcribe something, you have repressible or repressors bind to the operator, blocking off the RNA polymerase. So this is going to be a repressor. It's going to block RNA polymerase. Right? RNA polymerase can't move forward unless you either have an inducible system where the presence of some metabolite is going to cause RNA, the repressor to fall off, or you have something where you have a system where the repressor is inactive, so now you have the RNA polymerase that's able to go ahead and transcribe. And so now the presence of a certain metabolite will activate that repressor, and now that repressor will bind, and it's going to control gene regulation. So you have these, you can have inducers. Or co-repressors.
The promoter is Tata.
Promoter is Tata. And upstream of Tata is what? Yaka. Upstream of that, you got.
And but there's only like 10 base pairs or something between Tata and start codon, no?
Mm-hmm.
So the operator is very small region.
Very small region.
And part of that includes the Shine-Dalgarno system.
The Shine-Dalgarno sequence is on the mRNA. Well, it's technically found in the mRNA. I mean, the coding strand codes for it, right? But it's technically found on the mRNA.
Okay, but
I don't know. I don't know if it's a shared thing going. I know what you mean. Why there's so little
It
room.
only needs a few base pairs.
There's only
Yeah.
little room to have all these things going on. So maybe there's like a co-functional thing where it's the same sequence, a part of the same sequence. If you like do like a frame shift type of thing going on, where a certain segment will read for Sharon Delgarno, a certain segment will read for the operator. But the basic structure is that you have a promoter in between the operator in between the promoter and the genes. And then this right here is going to be the repressor, right? So this isn't a prokaryote. So now my question to you guys is, do eukaryotes also have polycistronic mRNA? Do we have mRNA transcripts that have multiple genes coded for them, on them?
That one it has a monocistronic.
So we mostly have monocistronic, but we also do have polycistronic, although it is rare. So one example, right, is nematodes. Nematodes are these worms, right? They're more basic eukaryotes, but they do have operons in them. But what's another example of polycistronic mRNA? Or how else can you get polycistronic mRNA? Through what mechanism? Adeno splicing. Trans-splicing, right? The trans-splicing can also help with that as well. So do you guys remember the basics, how, like, when is the lac operon operated? When does it function? When does it not function? When is it on? When is it not on? Another way of saying it. So what needs to be present for your lac operon to be functional? Normally your lac operon is off.
If you have the presence of glucose, sugar.
So if you have the presence of glucose, then you're going to turn this off.
Okay.
So it's in the name, right? Lac operon means like lactose operon. So it's going to create lactase. And so under the presence of lactose or a lot of excess lactose and minimal or no glucose, then you're going to have the lactose or allolactose, a form of chemically modified lactose, that is going to bind to the repressor. Once it binds to the repressor, it's going to cause the repressor to become inactivated. And this is going to basically allow RNA polymerase to go ahead and move forward. Right?
So the presence of sugar will.
Presence of a specific lactose in the absence of glucose.
Okay.
When you have lactose and glucose both present, it's like, because the reason why you prefer, you don't want to operate the system when you have both lactose and glucose present, is because glucose, or when glucose is present, is because this is only to break down lactose, and the reason why your cell is breaking down these sugars in the first place is to make energy, and so glucose is like the most energy efficient, the basic form that you want of sugar that you want to use to break down for cellular respiration, for ATP synthesis, right? And so, so glucose is like the main source. So if you have glucose present, you want to always use it. If glucose is not present, then you're going to do this lac operon mechanism where you're going to go ahead and synthesize lactase to break down the lactose. And lactose is a disaccharide made up of galactose and lactose. I mean, galactose and glucose. Sorry, galactose and glucose.
Yeah.
And so so if you break down lactose, you do get glucose. But the idea is why go through all that energy waste, right? Wasting all that energy to basically have transcription factor bind, have RNA polymerase do its job, have translation happen, and then do all of that when I can basically just use glucose and not have to go through all this gene activation. And so there's also a inverse relationship. So also for the lac operon, there's also another region before the promoter, and you guys know what that is? So it has to do with the lac operon, not really. It doesn't have. You don't have this in the TRP operon.
Cancer region or inducer or
has to do with the cap. So it's like the cap binding region. Cap site, you want to call it that? And so there's an inverse relationship between the presence of glucose and CAP. And CAP doesn't act by itself. We have CAP and CAMP working together. So when you have a lot of glucose present inside the cell, you have little to no CAP and CAMP present. And when the role of CAMP and CAP, they work together, right? Synergically. Where the binding of CAMP and CAP together, and them binding at this complex right here, the sequence, it basically helps to promote and recruit RNA polymerase. So it's also involved in the regulation of the lactic operon. So when glucose is present at high, you have less of CMP and CMP, or DC. Cyclic adenosine monophosphate. And CAP stands for catabolite activator protein, right? So both of these together go ahead and bind there when you have little glucose. When you have little glucose, the job of these two is to basically recruit or help recruit or make it more favorable for RNA polymerase to bind at the promoter. And we also said that if glucose is not present and lactose is present, allolactose, a form of lactose, will basically bind to the repressor, which will be bound to the operator and make the repressor fall off. That way RNA polymerase has clearance to move forward and make the lactase. Right? I want to quickly go over these questions, the last two questions. We only have a couple minutes.